Hello, I'm Dr. Ahmed Abdelal, and in this unit, I'm going to speak with you about the cellular structure and functions of the nervous system. So the human nervous system has many different anatomic and physiological divisions, and we'll start with the first part, which is focusing on the structure of the cells of the nervous system and their function. And the next two sections will focus on the structures of the nervous system as a whole. So we look at the structure of the brain, spinal cord, and the peripheral nervous system. But that will be in the next two sections. So the human brain, as you could see here, this is a 3 pound. The weight of it is 3 pounds. It weighs about 2% of your body mass. However, it consumes a tremendous amount of energy. 15 to 20% of the energy that you consume goes to the brain. Just as some fun facts, the nervous system in your body has enough electricity to make like a bulb for your fridge or stove, 40 watt. In babies, newborns, the brain consumes almost 50% of the nutrition that the baby eats. And of course, the brain also, comparative to the size of the body, is more. So when we look at the structures first, there are two kinds of cells, two categories of cells in the nervous system. First is the glial cells. And these are kind of called, glia comes from the word glue. And the glial means like sticky, and kind of they basically cushion the neurons. And then you have the cells, the neurons, which are more massive, they are bigger, and they actually, all of them, transmit and receive signals from different parts of the body. So the glial cells, there are many different kinds of them, and they have a tremendous number of functions. And without the glial cells, the nervous system cannot work. The neurons cannot function. So for every one neuron, there are about 10 to 50 glial cells. So together, you can think of it, I think of it like this. When, say, you order something online, and it arrives to you in a box that has all kinds of foams and things, cushioning it from all around. That the foam and the peanut foams and things that they put, these protect the thing that you received. So collectively, the glial cells have many functions, including insulation around the axons of neurons. So every neuron has, or most of the neurons have axons where they transmit signals across these axons to another neuron. So these axons, just like electrical wiring, they have to be insulated. And in order to be insulated, they have myelin around them. Segments of myelin will wrap around segments of the axon. So these segments, between every segment of myelin and another segment, there's a tiny, tiny gap between one segment and the other. And we'll discuss how this works later. But this is the myelin is made by two kinds of cells, two kinds of glial cells that we will discuss. Glial cells also provide structural support to the neurons. They provide nutrition to neurons. They regulate potassium and potassium levels, for example, in the extracellular space around the neurons. They contribute to the protection of the immune system. They make the immune system of the nervous system inside of the brain and the spinal cord. They also contribute to brain development in many different ways. Glial cells, one kind of them, makes up the blood-brain barrier. Just a small fraction of glial cells are able to transmit signals. However, the general, collectively, for the most part, they do not. In terms of neurons, there are many different kinds, more than 100 kinds of neurons based on their shape, anatomy, and function. And they tend to be larger in size, and they, of course, are less in number. And they transmit signals to each other, and they can communicate with glands in the body, like, say, sweat glands or earwax glands and so on. And they can also communicate with muscle fibers as well. The brain of a newborn has almost 200 billion neurons, and the brain of an adult has about 100 billion. And the reason is that even though the baby's brain is 1/3 the size of an adult's, it undergoes a lot of construction. And then after the construction is, as the construction becomes more and more completed, then neurons are discarded. And the neurons that that are used to connect and make networks, they become bigger and they need more space. So you end up deleting certain axons, deleting certain connections, synapses, and so on. So now let's look at just 5 different kinds of glial cells that are critical to the function of the nervous system. The first kind is a cell called astrocyte. Astro means star, cyte means cell. So the astrocytes, they are star-shaped, like this green cell here. They are star-shaped, and as you can see, they have many arms, and each arm has little feet. We call them pods. Like podiatrist is a foot specialist. Pod in Latin means foot. So these are pods. So the astrocyte, there are millions of them, of course. So an astrocyte communicates or has pods landing on a blood vessel on one end. And then on the other side, they have pods connecting on neurons, bodies of neurons, and different kinds of different components of neurons. So with the blood between the pod of the astrocyte and the membrane of the blood vessel wall, there is a special set of channels between, inside of every pod. And also the blood vessel has channels whereby the nutrition from the bloodstream is extracted by the astrocytes. It is extracted only the nutrition that the neuron requires, because the blood inside of the blood vessel has many elements that could be toxic to the neurons. So the astrocyte extracts the nutrients from the blood vessels and then processes these nutrients in the body and makes them refined and purified. And then on the other side, it passes them on to neurons. So without this, the neurons have no way of making, of having energy or food. So any malfunction in the astrocytes is going to mean that the neurons will be dead. So in addition, the astrocyte maintains, and again, I'm just speaking about the astrocyte as the kind, the type of cells. I'm not speaking that the brain has one astrocyte. We are speaking about millions of them. But if you study one, then you have studied the millions. So the astrocyte also has other functions. In addition to forming the blood-brain barrier and giving the neurons their nutrition, in addition to that, they maintain the potassium level or concentration levels in the extracellular space outside of neurons. So this will ensure that potassium levels, when they are too high, you will have probably areas that do not have enough. And also, that is going to be too much potassium is going to lead to less efficiency in the energy. So what happens is that the astrocyte will kind of will absorb the extra potassium. Potassium is needed for transmitting signals in and out of neurons. So if there's extra, the astrocyte will absorb it, and it will either keep it until the neuron needs it again, or it can pass it to on the other side to neurons that do not have enough in their extracellular space. So this way they regulate the concentration of potassium in the extracellular space. They also regulate, the astrocytes regulate the synaptic activity, which means a synapse is a point of contact between two neurons when they communicate together. We don't have one here, but I'll explain it later there. So these astrocytes regulate the synaptic activity by, for example, recycling some of the neurotransmitters. A neurotransmitter is a chemical element that binds onto a channel to help the channel open or close so that the signal can go through. And without a neurotransmitter, the cell cannot do its function. Most of the cells will not do their function. So the astrocytes regulate the level of neurotransmitters by also recycling some of these. When a neurotransmitter is, binds and then takes off after, then some of them will be broken down and discarded, recycled. And the astrocytes will help in that recycling process. Some of them will be used over again. But the astrocyte regulates the level of neurotransmitters by taking some of these and recycling them so they will not be, obstructing the communication. So here are, again, in addition, they provide structural support. This cell, for example, is holding on to the blood vessel here, holding on to more than one neuron there, holding them in place. So that is structural support. So now, these functions are very clear. They are expressed in concrete language, straightforward. So 1, 2, 3, 4, 5 functions of the astrocytes. Without these functions, the neurons cannot perform their job. So you could also see here, see how the astrocytes, they form the blood-brain barrier, and by connecting onto a blood vessel or capillary on one side, and then onto the neurons on the other side. And here's the detail you could see inside. There are channels here through which the nutrition, can be absorbed. Another kind is the Schwann cells. Notice the spelling SCH, Schwann. That's the name of a scientist who discovered that. Schwann cells form the layers that insulate the axon. They form the myelin. Myelin is the substance, the kind of fat that is used to insulate axons. And the insulation will enable the axon to transmit signals quickly so that that energy would not be lost along the way. The neuron that you could see here is a motor neuron. So it has a body, it has dendrites, it has an axon, and that axon has many segments. You could see many segments of myelin. The reason it is white here is usually fat is white. So you have segments of myelin. Each segment is one layer, is multiple layers of myelin made by one single Schwann cell. So the Schwann cell begins like this. First, making one layer, then continuing to wrap. And as it grows, it wraps more and more. So one Schwann cell could wrap up to 16 times, 16 rounds. And the level, of course, is going to determine how much someone can do. And if there is not enough insulation, that will lead to degenerative disease like ALS and Huntington's disease and so on. So you could see here the axon, and then the Schwann cell starts to grow around it, and then there's a cell body, and then that continues to wrap. As it continues to wrap, the cell body travels to the outer layer, and it continues to wrap until it becomes mature. But keep in mind that every Schwann cell insulates one tiny segment of a motor neuron. And these exist only in the peripheral nervous system. The peripheral nervous system includes all the nerves that are called the cranial nerves, and the spinal nerves that come out of the spinal cord. So Schwann cells only insulate axons of motor neurons, neurons inside the peripheral nervous system. These neurons can be anything, can be motor, sensory, or interneurons. The longest axon in the human being is almost 3 feet long, that goes from the waist to the foot. And it needs about 400, one single neuron. So again, the function of the Schwann cells is to produce layers of myelin to insulate axons in the peripheral nervous system. So then we have another kind, which is number 3, another kind of glial cells, and that is the oligodendrocyte. Oligo is comes from the term for a molecule of sugar. Dendro means branch, dendro, and cyte is cell. So oligodendrocyte, a cell that is, has branches like a kind of trees, and it is made out of a particular kind of sugar. This is the second kind of cell that provides myelin, but it provides myelin in the central nervous system. The central nervous system consists of the brain and the spinal cord only. Okay, the brain and the tail of the brain is the spinal cord. You can imagine it like a hard drive, and there's a wire cable coming out of the hard drive to carry all the signals and everything in the hard drive. That is the spinal cord. So the one difference between the oligodendrocytes and the Schwann cells is the oligodendrocytes exist in the central nervous system, in the brain and spinal cord. And one single cell insulates multiple neurons, or the axons of multiple neurons. As you could see here, this cell is insulating, providing insulation for one neuron here on one side, and then insulation for the other, and it can insulate hundreds of neurons coming around. So the layers here are less, maybe about up to 4 layers each. And in addition to providing the insulation, this kind of cell also provides support, structural support. So it will be holding axons from different sides coming in, and then that will make them kind of stay in place. So insulation and support. So I needed to know the difference between Schwann cells and oligodendrocytes, the two major differences that I mentioned. Another kind of cell is called the microglial phagocyte. Phagen is the Latin verb meaning eat, to eat. Cyte is a cell. Phagocyte is a cell that engulfs or eats other cells, which is basically a white blood cell that is, say, there's a bacteria, for example, here. This cell comes in, goes around it, and then until it circulates it completely inside, and then it swallows it, the bacteria, and kills it. So this kind of cell is a modified kind of white blood cell that lives inside of the brain and the spinal cord, and it becomes part of the immune system inside of the brain and spinal cord. So, for example, when someone has a brain infection, meningitis or encephalitis, there will be a lot of these cells, microglial phagocytes, because they will be fighting the bacteria or virus that invades the brain, and they do a spinal tap to draw fluid out of the spinal cord, and they measure how many cells per speck there are, and they can tell if there's an infection, because if there is one, there will be more of these. So these microglial phagocytes, they go around between neurons and around the brain and the spinal cord, and they clean up any debris, any destroyed neurons that are dying, or any debris that's there. They kind of swallow and clean it up, and they recycle dead cells. And also when there is an accident, trauma, a lot of cells die. They go around and surround the site of the injury and isolate it and close it off to provide as much protection as possible. So that is part of the immune system. radial glial cells or radial glia. These cells, they are like cylinders. So they're like cylinders, tubes that are open in each, both ways. So they connect with each other and they form long groups and long, what they call them, radius or radii. So long groups like this. Like the blue line. These, or you can think of them like hoses. And these hoses extend from the area of the neural tube where the brain cells are born at the beginning, for example, especially in early development when there's cell migration and proliferation. They extend from the site where neurons are born, deep in the brain, and they extend to the area where these neurons will stay there to function. So that this way, the new neuron is going to find its way. For example, maybe this neuron is going to be part of your eye, if your visual, mechanism. So it will be born in an area that is now known as the brain stem. But at the beginning, it's not quite clear. We're speaking about like 6 or 7 weeks of pregnancy. So this little hose guides the newly born neurons, and they cling onto it as they move on to the area where they are going to be implanted and they will function. They will get the nutrition as they keep going. They will be nourished along the way. So while they are traveling, they are growing and they receive nutrition from the inside of the radial glial cells. So, and when they reach their destination, now they become mature and they can function and they can be embedded into pathways and so on. Without this migration, any damage to the radial cells or malfunction, that will disrupt cell migration, and that could result in a lot of disability and brain damage. So now let's look at the neurophysiology and the neuroanatomy and the neurophysiology of the neurons. So now we needed to understand and to really review and study the 5 kinds of glial cells that we have discussed. Straightforward, clear, know the structure, know the function. Now we'll speak about the neuroanatomy, means the structure, physical structure of the neurons, and their neurophysiology, means the function, how do they work. So in terms of function, physiology, there are only 3 kinds of neurons. 3 kinds. A neuron can be a sensory neuron, can, like, say, your hearing is the signals are transmitted along neurons that do not move, but they just fire electrical signals. Sensory neurons. Your vision, when you look, there are neurons that will take images and enable you to see without necessarily requiring movement. Of course, in vision there are movements neurons that are motor neurons that enable you to move your eyes up and down and sideways and so on. But there are also sensory neurons that enable you to channel images and to see things. So sensory neurons, for example, all your entire surface of your skin, inside and out, is lined up with sensory neurons. Like, how do you know when someone touches you? How do you know that someone just touches a hair on your skin? That is because of the sensory neurons that line up your every, part of your skin. And then that transmits signals that go along your arm to the spinal cord, to your brain. And then we have motor neurons. These neurons enable us to move. They move different parts of the body inside and out. And then we have neurons that connect sensory neurons with motor neurons. By the way, we can call these primary sensory neurons, or primary motor neurons, or sensory neurons, motor neurons. And then the neurons that connect both of them are the interneurons, neurons in between that connect the two together, because you have to connect sensation to movement. Like, say, I feel something crawling on my arm. Immediately that triggers action. If that thing is going to sting me, I'm going to withdraw my hand. So I have to connect my sensation with my activity, my motor activity. And the only way we can do this is with the interneuron. And the connection is for mostly in the spinal cord. So I give you an example here. This is an example of a sensory neuron. You have a cell body. On one side, you have one pole. You have a one that is has an axon here. Continues. The axon continues here. So let's say this is on your skin, embedded in your skin, the receptor cells, or maybe inside of your nose to make you smell. So these are the receptor cells. And then they conduct the signal when they fire. The signal travels along the axon, goes by the cell body to be processed, and then it continues along the axon on the other side. And then it goes to dendrites to be transmitted to another cell. So that is an example of a sensory neuron. The next to it is an example of the motor neuron. So here's the cell body. There are dendrites, sometimes called dendria. And there's an axon, one axon. And after the end, there are synaptic terminals, that are called sometimes telodendrites. But synaptic terminals are more common. And the very end of this, you have little things like little feet called synaptic pods. So motor neuron. And here you have another kind of neuron that is where you can see the cell body and the axon, and then you have the branches. This is called the pyramidal neuron. That is a kind of this kind and this kind. They're called multipolar neurons because you have a cell body and you have big branches here. Each one begins as one piece and then it branches off on one side. And then on the other side, you have one axon and then synaptic terminals. So that's called the multipolar neuron. See, 1, 2, 3. And here, there's another example of multipolar neurons. So the dendrites have branches and they're big and they branch out on one side. There's a body, and then the other side you have the other pole on the other side. So these are interneurons. These are kinds of interneurons that connect sensory and motor and sensory neurons. Let me explain, for example, the circle here, the circuit, or the kneecap reflex. And you need to understand it, and you need to know how to explain it step by step, just as I'm explaining it now. So here, you have a cap that is connected to a tendon of a muscle called the quadriceps muscle. Yeah. And then out of that, you have sensory neurons. They just put one single neuron, but it doesn't mean it's only one. There are many of them, but we just use one as an example. So the sensory neuron here is connects with the muscle fibers of this muscle. And so you have the dendrites embedded into the muscle fiber. And then the axon here, the axons, continues to travel. And then here's the body goes as the axon approaches, in the trunk. Then it goes, as it goes into the spinal cord. This is a slice of the spinal cord. So we are speaking about this here. It enters the spinal cord and it synapses with an interneuron. So the ends here, synaptic terminals of this sensory neuron, synapse means connect with the dendrites of the interneuron. And that's inside of the spinal cord. Then the dendrites of the spinal cord, of course, continue with the body of the interneuron. And then the signal travels from there to the axon of the interneuron, to the synaptic terminals of the interneuron. And all of this is happening inside of the spinal cord. Then the signal goes from the interneuron, the synaptic terminals, to the motor neuron. There's another connection that the sensory neuron has directly with the motor neuron also. And that means that the signals coming from the sensory neuron activate the motor neuron. So then the motor neuron is going to cause these muscles to contract. When a muscle contracts, it will shrink. It will shorten. When the muscle shortens, it is going to pull with it whatever is connected with it on the other end. It will pull the whole leg up like this. And this is just one leg, and it shows you the movement. It goes from here to here. So that is how the knee reflex works. Again, I'll explain the steps. And you need to know them in sequence, because if you do not know them in sequence, they will not work. The reflex will never occur. So when the tendon at the knee, below the knee is tapped, that is going to activate the sensory neuron in the quadriceps. The sensory neurons will transmit signals that will go through to the synaptic terminals, to their synaptic terminals here at the end. And these synaptic terminals connect with the interneurons inside of the spinal cord, that's interneuron, that little green thing. And also with the motor neurons at the same time. So the connection between the synaptic terminals of the sensory neuron and the dendrites of the interneurons, they carry the signal to the dendrites of the interneuron. From there, the signal travels to the dendrites, the synaptic terminals of the interneuron. From there, the synaptic terminals of the interneuron transmit the signal to the dendrites of the motor neuron. And then the signal continues along the axon of the motor neuron to the muscle fibers, where at the neuromuscular junction between the motor neuron synaptic terminals and the muscle fiber, that will release neurotransmitters that is going to contract the muscle to make it shorter. When that muscle shortens, it will pull with it the whole leg. It will pull it forward. Now, why the neurologists have, or your physician will do the kneecap reflex? So in terms of anatomy, in terms of what they look like, how are they big? Are they small? Do they have one pole in one end, another pole? Like in terms of their anatomy, neurons come in more than 100 different kinds. And they also have different functions. So most neurons have the basic structure that is similar to the motor neuron. This is why the motor neuron has this, as having the simplest kind of structure, anatomic structure. It is used as a model generally for all neurons. So when you study it, you have basically studied the structures of other neurons. However, other neurons will have additional features or might have less features. So let's look at the anatomic parts of the neuron. First, we have a cell body. That is the most critical part of the neuron, a cell body. And inside of it, you have a nucleus, cells, and can reproduce materials that are needed to maintain the health and integrity of the whole neuron, to produce spare parts, for example. So in the cell body, also you process signals of the neurons. Out of the cell body, there are little extensions known as dendrites or dendria. So these dendrites are the site. Their function is to receive signals from other neurons. So every dendrite has many, many areas that are shaped like this. And then synaptic terminals from other neurons come. They have an extension like this, like a foot, synaptic pod, and they come and kind of come close to the dendrite, and they make a synapse like this. There's a little gap in between. So dendrites receive signals. Dendrites never send signals. They receive signals either from your skin, for example, or from other neurons. There's a little triangle that is at the end of one of the cell body. That triangle, that is an extremely important part of the neuron. That is called the axon hillock. The axon hillock is the site that processes signals that are coming into the neuron. If these signals are 500 signals, 5,000, 10,000, 50,000 signals, they are all processed inside of this axon hillock. So the axon hillock determines if the neuron is going to fire a signal or not. So it will examine all the signals coming through, and if, say, 500 signals say fire, 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 and then 300 signals say don't fire, don't fire, it goes with the more signals it's like a democratic process. With with the more signals that tell it to either fire or not to fire, it will go with the majority. So that is the axon hillock. It connects the body of the neuron with the axon of the neuron, and again, it determines if the neuron is to fire a signal or to not fire a signal to the next neuron. And then we have the axon that is like a little thread kind of, or wire that connects, that goes between the axon hillock and basically it ends with synaptic terminal, synaptic branches, and these when signals come from the along the synapse, they can be transmitted, copies of the signals will be transmitted to different neurons. So keep in mind that a signal in a neuron only travels in one direction. It cannot travel back. The signal always comes from the cell body, the axon hillock, through the axon to the synaptic terminals. It does not travel back. So the synaptic terminal then connects onto different neurons, different areas, and then the signal goes from the synaptic terminals to other neurons. So because of that, let's look here. There are, or I'll explain it when here. Here's the synapse. So you need to know the structure of the synapse, how the synapse is made, the anatomic structure of it, and how it functions, how it works. So first, this is a synaptic terminal coming from one of these areas here. At the end of a synapse, at the end of an axon. So synaptic terminal, or you can be specific and call this a synaptic pod. And on the dendrite, sometimes on the cell body, on the dendrite, there's a particular area that is curved, and it just, the synaptic terminal fits within it, and there's a gap in between them. That's called the synaptic gap or the synaptic cleft. And the two really do not, that space is always in there, most of the cells. So inside of the synaptic terminal or the synaptic pod, there are little vesicles, almost look like bubbles. They have 3 layers. Each bubble has like 3 layers. And inside of these are neurotransmitters. And on the other end, the cell that receives signals, on the other end, the membrane of that cell has, of that neuron, you can call it cell as well, it has channels. These channels open and close to take in particular elements, particular elements that transmit the signal, or to close the gate. So these neurotransmitters, when they are released by the synaptic terminal, they are released into the space, into the synaptic cleft, and they dock on the surface of a receptor channel of the receiving cell. When they dock, they can either open that gate, that channel, or close it. If the neurotransmitter opens, that is called what do you call it? An excitatory neurotransmitter, and it will produce an excitatory action. It will make the signal go through. If that neurotransmitter binds and causes the cell to close, the gate to close, that will become an inhibitory neurotransmitter, and that signal will be called an inhibitory signal. So lastly, the this is part of the neuron that is sending a signal. Remember that a signal can only travel in one direction. So when it comes, it only has to go from the synaptic terminals of a neuron to the dendrites or to the cell body of another neuron. So synaptic terminals always pass on signals. Dendrites always receive signals. So this neuron here, this dendrites are receiving signals. The signals will travel along it to the cell body, to the axon hillock, to the axon, and to the synaptic terminals. And from there, this same neuron will be sending the signal to another neuron. So this neuron is both receiving and sending. So on the other end, if you put a neuron, it will be just like this one, but the dendrites will be communicating with the synaptic terminals here. So the same way, signals travel in one neuron the same way. So we call this kind of neuron, or this part of the neuron, we call it the presynaptic neuron. The presynaptic neuron. The signal that it is bringing in is called the presynaptic action potential because it is coming from the presynaptic neuron. This neuron that is receiving, or the part that is receiving the signals, is called the postsynaptic neuron. Imagine like the synaptic cleft is the boundary, is the actual communication site. So this is in front of the action communication site, in front of the synapse. So we call it the presynaptic. And this one is behind the synapse. So when we say the presynaptic, automatically it means the one that is sending in a signal. When we say the postsynaptic, automatically it means the one that is receiving a signal. So again, the signal comes along as the electric firing comes along the axon of a neuron and goes into the synaptic terminal. It causes the vesicles that carry the neurotransmitters to come close to the membrane or the wall of the synaptic terminal. And then as one vesicle comes and touches, then you have little tiny hooks called synaptic snares that come out and clutch onto the vesicle. They take it, stick to it, and then bring it and pull it to make it touch the wall. So when it touches the wall, then it makes its wall, the part of the wall, fuse with the wall of the membrane of the neuron, of the synaptic pod. And then as it continues, then that fusion then as it becomes part of it, it opens up on one end without exposing the inside of the cell to any other elements that come from outside. So it's an ingenious way of dumping out the neurotransmitters without having to break the wall of the axon terminal. So the little neurotransmitters, they will go around and they bind on the outside of a receptor channel. And when they bind, they serve like a lock and key. They bind and they either cause the channel to open, to take in calcium, potassium, and so on, or to close to prevent the signal from going through. So this is how synapse works. So you need to know the parts of it, the anatomic structure of it, and you need to know the functions, how the signal goes, how the neurotransmitters work, and how they bind. After the neurotransmitter does its binding and allows the channels to open or close, then it takes off it just floats back in the extracellular space. Some of the neurotransmitters are recycled, broken down by the astrocytes, and some may be used again, maybe reuptook. See these channels here? They are called reuptake channels. They can reabsorb the neurotransmitters back in again to be used again. So, and some also are kind of taken and broken down, either recycled or used over. So again, the neurotransmission process, it means basically a description of the steps that the signal takes to go from one point to the other, from point A to point Z. So first, we'll begin it from the postsynaptic, neuron. That is the neuron that is going to be receiving a signal. The postsynaptic neuron, through its dendrites or directly through its body, it will receive a signal from another source, from another neuron, or directly from the environment, like someone touching your skin, or something pricking you or stinging you, for example. So the signal comes either from the environment or from another neuron, and it is received either by the dendrites, by the cell body, or even by different parts of the same axon. But the signal then goes to the cell body to be processed, and it goes to the nucleus to be processed, and then it goes to the axon hillock. This is where the decision is made whether that signal should be fired or not fired. When I'm presuming the signal will be fired, that means it will travel across the axon. And from there, it will travel to the synaptic terminals. And from there, it will be broadcast to other neurons. As simple as that, but you need to know the steps and explain them clearly. So what happens to how does the neurotransmitter binds again, and how it works, what happens to it? First, when the neurotransmitter is released, it is released to the synaptic cleft. And if the signal is, or the neurotransmitter itself induces an excitatory signal, it means it opens the channels, the receptor channels, and it allows the signal to go through. If the neurotransmitter causes the gate to close, that means that the signal is inhibitory, and it will prevent the signal from going through, which means that the other neuron is not going to fire in response. The firing is called the action potential. So the action potential is the spark, the electrical spark, that electrical firing that is going to be produced is called the action potential of a neuron. And this is called an all or none event. It means it either occurs or it doesn't occur. There is not a baby action potential and the potential. No, it either comes, the neuron, it either causes the neuron to fire or not to fire. It doesn't come like in different sizes. So that's what we mean by the action potential. There is some kind of almost like a trigger, kind of pull, you reach a point, and then if you reach that point exactly, the neuron will fire. If you do not, then the neuron doesn't fire. But that is an all or none event. It means it either occurs or it doesn't occur. It doesn't mean that there are baby action potentials or big ones. So now we're going to speak about the development of the human nervous system. We'll speak briefly. That's not, I teach an entire course on the development of the nervous system. So, but this is just a very quick highlights. The neural tube, the first, after fertilization, you have one single egg called the ova. And then when it is fertilized, the sperm of the male comes in and goes inside of the egg, and the egg closes. And then the sperm has 23 chromosomes. And then once it goes inside, the skin around, all the structures around the sperm will be destroyed, and then it will open up the DNA and just throw it inside of the ovum. So now we have 46 chromosomes. Now we have a human cell instead of just 2 sex cells. They unite into one. That cell then starts to divide. And then as it keeps dividing for over the first 3 weeks of pregnancy, then you have 3 different layers. You have like a ball that has, or a balloon that has another balloon around it and another balloon around it. You have 3 layers of cells, just like this. Once the 3 layers are formed, then something happens. You have on the back of the fetus or the embryo, you have like a little indentation on the back, like a little valley starts to emerge, and that becomes deeper and deeper and deeper, and then when it goes deep, the 2 edges of it, the top, will meet each other and they will fuse. Now they have formed a tube. That's called the neural tube. This whole area is called the neural crest. In this area, on top of the fetus, on top of the embryo, in that little area here in the neural crest and the neural tube, this is the site where all neural cells are born. They are produced, but they have to be shipped out to the sites. You have to send neurons to different parts of to form the frontal part of the brain, the visual, centers for vision, centers for memory and hearing, centers for decision making, motor, all places inside. And the neurons travel all over the body to make the nervous system. So that is cell migration. So first, we have proliferation, the production, very, very rapid production of neurons. Remember, that new baby has to come out with 200 billion neurons. So it requires a great number of neurons to be born at the rate of 250,000 every minute. 250,000 cells are born every minute. So these migrate until they populate the different parts of the nervous system. And I explained how the cell migration, in many cases, especially to the distant sites, how it is guided by the radial glial cells. So that is in a nutshell how this process goes. It is completed by the end of the 6th month. That is where the fetus, the nervous system is kind of complete. It's not, of course, mature. It needs more, but the number and the population of cells, all of the cell migration process is completed during the 6th month. So now let's look at a few basic concepts here. Nerve, you know what a nerve is. You hear a lot about a nerve. But the nerve is basically a rope-like bundle of axons that travel together in one direction. So, or specialize in one thing. So this is a nerve. and it comes and you have bundles and bundles of axons. These axons specialize in the same function. This could be, for example, this could be a nerve like the auditory nerve, for example. So all these axons are bundled together into that cable, and with them you need nutrition. So you have little, blood vessels coming through. You have also a wrapping around these bundles, tough wrapping that is to protect the fibers of the neurons. So that is a nerve. A nerve consists of bundles of neurons that play the same role or have similar functions. The gray matter refers to the very top layer of the brain that is called that's called the cerebral cortex. It is the word cortex comes from the tree bark. So the outermost layer that surrounds the entire brain and the entire spinal cord, that is called the gray matter. However, in the spinal cord, this distribution of the gray matter and white matter are different. But the spinal cord does have gray matter and white matter. But for the brain, the most of the gray matter is on the outermost layer that is known as the cerebral cortex, that tiny 4 millimeter layer that wraps around the entire brain. And it is gray because it has a lot of oxygen, a lot of glucose, and it has bodies of the cells. And that is why it is rich with these nutrients. Then out of all these bodies and also glial cells, and then every neuron is going to send down an axon. Remember, the axon has myelin, and these axons all go down into like fibers and tracks and all of this. And because of all of this myelin, that is white. And this is where the name white matter comes. So the white matter is basically all the axons coming from the bodies of the cells. The greatest majority, 80% of the cell bodies, exist inside of the cerebral cortex. There are some cell bodies that exist deeper in the brain to form areas that are called the basal ganglia and other areas that we will speak about later, and the brainstem and so on. So, but that is when we say white matter, it is the cerebral gray matter is the cerebral cortex of the brain. And when you say white matter, is the underlying area beneath the gray matter. And it is white because it has myelin, a lot of myelin. So white matter, gray matter, fasciculus is a kind of pathway or a tract. And we'll study some of these. And neuromuscular junction is the point where neurons, the synaptic terminals of neurons, synapse or contact muscle fibers, and they form special channels where the axons, the synaptic terminals of neurons, release neurotransmitters to activate or to cause muscle fibers to move, and muscle fibers have that ability to move in response to electrical stimulation. This is one hemisphere of the cerebrum, or one hemisphere of the brain, the other hemisphere, and the point in between them, that bridge, is called the corpus callosum. This is one view of it, superior view. This is a coronal view. If you cut it this way, coronal view. then you can see this one part of it here, corpus callosum, corpus callosum. So it connects the right hemisphere and the left hemisphere. Okay, so what will follow then is part 2, and I hope that you found this session well detailed, and that it provided you with enough detail to have a good understanding of the cellular and cellular anatomy and physiology of the nervous system. And the next segment is going to focus on the structure of the different parts of the structures of the nervous system, focusing on bigger areas like certain areas in the brain and also the spinal cord and the nerves and so on. And the third one is going to focus exclusively on the cerebral cortex and the cranial nerves, and that will end the unit. So thank you.